This is part 2 in our series about evidence for the earliest parts of Earth history, geologic time, how maybe life came to be on Earth in the first place. So the first part of the video kind of focused on the formation of the Earth, formation of even the universe, everything that exists. This one's going to focus much more closely on how we know, or what kinds of things we might know about earliest life on Earth and how that sort of came to be. So this is a little bit more biology than the other one was. The other one was a lot more geology, I guess. So in review, the last video we talked about kind of some of that early Earth history, that the very first years on Earth were incredibly unpleasant. They call it, like Hades-like in terms of what it was. Lots of molten rock, very little land. The crust hadn't even formed yet. But yet, as the Earth sort of cooled down a little bit over a billion years or so, that it started to rain a little bit, and it started to form little lakes and ponds. Still had an unbreathable atmosphere as far as we would be concerned. Didn't have a great atmosphere in terms of ozone layer, no protection from ultraviolet radiation, had lots of huge storms with lightning, huge temperature extremes, again, without much of an atmosphere. It was really, really hot during the day, really cold at night. Definitely not a great place to live, but somehow this is how we think life actually got its start. At the very beginning, of course, it wasn't life like we think of it. It was basically just sort of interesting chemistry, but somehow this formed that very early place, sort of that cosmic primordial soup, so to speak, that could have placed the right things in the right place at the right time to somehow get living sort of biochemistry going. So this primordial soup hypothesis, I keep joking about it. There were two scientists back in the 20s named Oparin and Haldane, and yeah, you should probably know those names, that had a theory, a hypothesis for how they thought life could evolve from non-life, or how biochemistry could happen from non-biochemistry. Their idea was that if we had these correct ingredients, nitrogen, hydrogen, carbon dioxide, water vapor, plus a lot of energy, that somehow organic molecules could form, as long as oxygen is not present. And that's a critical idea here. If gas oxygen, O2, is present, this chemistry doesn't work at all. And so it's really a good thing that that atmosphere was different than it is today. So if we can start to get those chemicals kind of getting together, put them under energy, we can somehow get things like biochemical molecules, like really early amino acids, really early nucleotides, really early even sugars, or sugar-like kinds of compounds. And if these molecules can form either in the air or in the water, that with rain they could accumulate in that ocean. And the ocean in those days was really, really warm, and that's where that idea of primordial soup came into play. The ocean was quite a bit warmer than it is today. And then if you give those chemicals, those pre-biology chemicals, enough time to simmer, and we're talking about millions and millions of years here, that eventually those molecules could combine into more and more complex types of compounds, again, like proteins, or like nucleic acids, or like carbohydrates, kind of on their own. Again, it seems like that's not possible, but we're talking about a lot of time. Millions to billions of years for chance events to occur is plenty of time for chance events to occur. So this was confirmed. There were some guys that came along a little bit later in the 1950s named Yuri and Miller, and they came up with this sort of crazy-looking experiment sort of thing that they were trying to test that Oparin and Haldane sort of hypothesis. Can we actually do that? So you're telling me that if you stick carbon, hydrogen, some water vapor together, and we zap it with electricity, that you can get life compounds? Yeah, right. Well, that's what they did. They had a kind of a flask, I don't know which one to call that, a big vapor zone up here that's got the correct types of gases that we saw in that early atmosphere. They ran a bunch of electricity through it, sort of simulating what we'd get either from ultraviolet radiation or from lightning strikes. And then we let it kind of run through the system. This is a condenser cooling it, kind of like what we would get if it rained, for example. Kind of cool it down, get the water vapor to come out. And then down in here, in this bottom part of this region, you get this soup. It's a type of water, basically, that's got organic compounds in it. They actually found, and I love this phrase, red goo. I didn't do that highlighting, but there's red goo that formed in the bottom of this. They were surprised to see this, that it actually had amino acids in it. They could actually then heat it up, get the water compounds back, and then kind of rehydrate it if they needed to. So they had this big system. And so they actually did decide, or did confirm, that from non-living chemistry, methane, ammonia, water vapor, and hydrogen, these are non-organic compounds, that if you run enough electricity through them without oxygen present, that you can actually get those building blocks, amino acids, pre-nucleotides, and a variety of other things as well. So this has been sort of confirmed that you can get organic chemistry from non-organic chemistry under the right conditions. We've also found that this has not just happened on Earth. It's actually kind of surprising to think that some of the things on Earth may have come from space. They're not life. We didn't have aliens, but maybe there were some compounds that came to Earth on the backs of meteorites that actually contained some types of organic compounds that we really don't see here on Earth. And so you can see down at the bottom here, amino acids, some identical to ones that we've seen on Earth, and some actually not found at all on Earth, completely alien amino acids, and nucleobases, which are kind of like nucleotides, they're really close to that, have been found in space. And so it is possible that some of those compounds did come here on early Earth and sort of added to that soup and helped kind of spur it along, or maybe helped it to go faster than maybe it wouldn't have otherwise. But life itself didn't come from another planet, but maybe some of the compounds that supported life could indeed have come from other planets. In terms of the overview of what this current idea is, called the Heterotroph Hypothesis. This is that current idea of the primordial soup and how did we get to life from the primordial soup. You kind of talked about this first kind of couple of sections on here, that somehow we had inorganic molecules like hydrogen, water vapor. We know those were here. They can sort of start to create that soupy bit where you start to get really simple sugars forming kind of by themselves in the ocean. Complex organic molecules require some energy, and that would be like lightning strikes, ultraviolet radiation, a variety of things like that, just the heat from the planet itself. And you can get this buildup of these more complex kind of molecules that you've heard us talk about in class before. And then we get these last three, coacervates, is this word, protobionts, and primitive cells. These are the last three kind of big steps that need to be taken to get from chemistry up here to almost biology. Coacervates are almost like cells. And so we'll kind of talk about what those next steps are here in the following slides. So a coacervate is kind of a crazy looking word, isn't it? Coacervate's what you get. It's like a bubble of chemistry, if you want to think of it that way, where you've got lipids. Remember, we've talked about lipids don't like water, and so if you've got lipids together, they'll start to form these bubbles, kind of like pre-membranes almost, that can capture chemistry on the inside. And both of these are pictures of coacervates that were formed in a lab, but under kind of those early Earth sort of environments. And so it's possible that some of those chance bubbles could have formed with just the right mix of chemicals inside, that chemistry got captured. And that's what we're talking about in this bottom paragraph here, that you could get repeatable sort of self-sustaining chemistry that's almost like a type of metabolism. And if you can capture that in there, it's almost like a little cell. It's not a cell yet. It's not even a protocell, but it's kind of getting along those lines. And if you can get those self-sustaining chemistries to repeat themselves, to be able to replicate, then you're starting to talk about that next step, getting one step closer to life. And that's what we would call a protobiont. A protobiont is that next step where not only does it have some sort of a membrane, an outside versus inside, it's got some sort of chemistry going on the inside, a metabolism, but now it's got a reproductive capability. It can maintain itself for generations, not just once accidentally and then be gone or break or burst or whatever, but to actually make generations of it itself. We're not quite sure if the reproduction came first or the reproduction came second, but somehow they had to have combined by chance, again, over these billions of years that we're talking about, to potentially create some sort of a reproductive pre-cell. Again, protobiont means almost living thing. That's actually what that word means. So somehow in here we had to have RNA, and I'll talk about RNA here in a second, but some sort of a nucleotide. We talk about DNA today. Well, we're pretty sure it was RNA in these early cells, and then some sort of chemistry occurring, and if they come together just right, you get a cell that looks kind of like this, or a protobiont, not quite a cell, where it's got the membrane on the outside, little bubbles of chemistry on the inside, and some sort of a DNA molecule, a self-replicating molecule, to kind of keep it going year after year, or week after week, or day after day. As I mentioned earlier, we think it wasn't DNA that started it. We think it was RNA that started the whole thing for a variety of reasons, but it's called the RNA world hypothesis. DNA, I know now, is the big deal, but we're pretty sure that RNA started it. RNA is simpler structurally. It's only got, one side and the nucleotides. It can deal with ultraviolet radiation a lot better than DNA can. It's also capable of acting by itself. It acts like an enzyme, so it can actually make chemistry happen. And so you can kind of see in this little diagram here that RNA can assemble, we've done it, that it can assemble itself by chance from inorganic sources, little raw materials. And there's two different things that RNA can do. Step number 2, RNA can make more of itself. Just like the process of replication you learned about in Unit 10, RNA can make more of itself. Ribozymes are little, basically RNA type of enzymes that do jobs. But RNA can also make proteins, and we call that transcription or translation today. But it can also take part in doing that job as well. So it can do all the things that life would need it to do. And somehow, at some point in time, getting that wrapped into a nice solid membrane, a nice double phospholipid bilayer, can actually provide the protected environment that DNA needs. And why would cells go to DNA? It's more stable. It can last longer. It can be a much bigger molecule. It doesn't break down over time. But because it's got the two sides, it's not functional. You guys should know this from Unit 10, that DNA itself doesn't do anything but hold information, just like a book doesn't write a report for you. RNA is what actually does the report writing. It's actually what turns that stuff into information, proteins and things along those lines. So it took time to make that particular transition. So we think that RNA was kind of the beginning of this. So again, now you've got those things that you need for life. You've got a lipid bilayer, a bubble. You've got chemistry going on the inside, it's a metabolism, and you've got molecules like RNA and eventually DNA that can allow a cell to kind of maintain information and to be consistent generation after generation. This little slide here, I would say hit pause and take a chance to read it. It's actually from Time Magazine when they did a big story about this. But it kind of chronicles that idea. It puts it in a little bit nicer form than I can in terms of how is it possible that we could go from that really early kind of pre-cell to a protobiont. It talks about a little bit what ribozymes are and things like this. So this is a good time to hit pause, take a look at that, and this really describes that transition from RNA to DNA, and then from DNA to protein. And that's that key point that now, as living things today, we are governed by DNA and proteins, and RNA do the work. In the original days, it was RNA doing all the good stuff. Okay, just about the last idea here. Want to make clear that when we're talking about all this kind of very hypothetical kind of model stuff, these are theories that have been built on a variety of lines of evidence. There isn't just one thing that we're sort of guessing as how this is. And this little slide here kind of shows a variety of the different types of evidence that we use to kind of build our ideas about what early Earth was like. We've got evidence about the atmosphere, that we've got a drop in certain chemical compounds and eventually a rise in oxygen. This is where photosynthesis came into play. It was much later. Remember I talked about the Hadean period? That's that really nasty volcanic period in Earth's history. Archean is the later Earth's history. We've got the idea of isotopes. We've got fossils. We've got biomarkers. These are actually proteins and chemicals like that. And then we actually have geologic record. We've actually gone and dug up particular kinds of rocks that give us examples for this. So there you have it. Here's your big ideas from today's slides. Ideas about the Big Bang Theory and redshift, about formation of the early atmosphere. Some ideas about the formation of the Earth itself. That idea that oxygen-free had to be a critical part of it. We've got two major pairs of scientists that collaborated to give us some ideas. We've got coacervates and protobionts and the RNA world. See you in class in about 5 minutes. Bye.